Thank you very much for registering and joining the webinar. My name's Scott Pattison, Senior Director of Business Development at uBriGene. I've been with the company a little less than 2 months, but very exciting to be part of this group. Amazing technologies, amazing team, amazing culture, amazing client-focused business model. So I'm thrilled about the future of uBriGene, and a big part of it is our team and our technologies. And one of those key members of the team is our Executive Director of Strategic Business Development, Ken Warrington, who's going to lead the webinar today. Before we jump into that, let me just remind you about some of the logistics that we have on that opening slide. So everyone's going to be on mute. We have a Q&A box, chat box over to the right of your screen, and invite everybody and anybody to put comments and questions into that chat box. We'll wait until Ken's done with the presentation to address those questions, but we will get to them. So thank you for that. And then before Ken gets started, I have a very quick one-question poll. So I'm going to bring that up and give you guys about a half a minute to submit your responses to this poll question about what grade of lentiviral vector production you might be needing. So I'll give everybody another 15 or 20 seconds to pick their button of choice, and then we'll dive in. All right. Doesn't look like everybody has voted. If you can see the poll question and can vote, please do so. I'll give you another 10 seconds. All right. Thank you very much for participating in that. That's very helpful to our marketing efforts and bringing up high quality webinar materials for future presentations. So let me introduce Ken. So Ken is a terrific CGT expert. He has very, very broad experience, everything from discovery through GMP manufacturing, IND enabling preclinical work, all the way through to early clinical program execution. Ken has a decorated career in both in academia as well as on the commercial side. So he served on the faculty at the University of Florida for many years, and he served on various companies in the cell and gene therapy space, focused on contract manufacturing and contract testing. And so without any further hesitation, Ken, feel free to take it away.
Thanks, Scott. Appreciate the kind introduction, and certainly welcome to the team. We look forward to working with you as we look to really focus on basic education programs through our webinar series to help level the field, if you will, in terms of knowledge base as we introduce our various cell and gene therapy platform offerings. Today, and thanks everyone for joining. Today we're just going to cover some lentiviral vector basics at a high level. I'll introduce our platform, how we can enable clients through access to our cell lines, plasmids, and lentiviral services, and then provide a high-level example of how the lentiviruses are used in cell therapy applications as an introduction to our new facility that's coming online in Maryland, our Center of Excellence for Cell Therapy. Just a brief high-level introduction. I won't go through all of the specifics other than to say we bring a pretty broad catalog of offerings to the cell and gene therapy space, from hundreds of GMP plasmids manufacturing campaigns executed, a number of vector platforms, including lentiviral vectors. And obviously, these can be used in a broad number of cell therapy approaches. And then, to supplement all of our manufacturing activities, we do have in-house all QC release testing and a process and analytical team that can support process development, assay development, and qualification to support the cell and gene therapy, as well as our brand new offering in the RNA space, focused initially on mRNA and circular RNA. But also aggressively pursuing additional RNA technologies such as self-activating, self-amplifying. So let's talk a little bit about lentiviral vectors. And at uBriGene, we work with a pretty standard platform in terms of the third generation and 4 plasmid platform. Typically, client brings to us their vector or shuttle or transfer plasmid, whatever you want to call it, and we can support the production of a GMP batch of that plasmid to plug and play into our, if you will, into our platform offerings, plasmids, GMP, qualified plasmids off the shelf. That include the packaging plasmids, obviously Gag and Paul, involved in delivery of reverse transcriptase and integrase, the REV, which is a bit of a regulatory packaging plasmid. And then a lot of our development has been with the envelope plasmid VSVG. But obviously, this is another area of plugging an alternative glycoprotein into your lentiviral production. And certainly this platform is well characterized as the field now moves to looking at additional moieties, be it plasmid or additives to the media that have the ability to increase both yields and titer. We work in our own proprietary 293 T cell suspension, but we also have adherent capabilities, and I'll talk a little bit about the development of our suspension-based platform. And then just a brief cartoon here on the right, really demonstrating the reason we use lenti in cell therapy applications in particular. Lenti is an envelope virus. Obviously, it has a large packaging capacity relative to some of the others out there, about 9 to 10 kb. But after events at the cell surface binding to a specific receptor, the lentivirus will fuse. Deliver both reverse transcriptase which, since lenti is a single-stranded RNA virus, generates that viral cDNA, and the key component of deploying lenti in cell therapy applications is, of course, its ability to integrate, delivering stable long-term gene expression. This can have some caveats, obviously. Lenti does have a preference for integrating in transcriptionally active genes. So there have been examples, at least maybe not direct causes, but certainly examples now that the FDA has observed with the ongoing CAR T programs where this can lead to subsequent malignancies if the integration takes place in an oncogenic situation. But ultimately, since these are non-replicating vectors, that we would, from a vector standpoint, it would stop here. But certainly the rest of the life cycle includes all the additional viral protein synthesis required to form the viral RNA packaging within the capsid assembly and ultimately budding and maturation at the plasma membrane. So in general, our platform flow has been developed with the 4 plasmid system. Obviously, just again as a reminder and at a high level, we go through, we have single-use technology, so expanding over 293 T cells, followed by transfection of those in the reactor, and then a pretty straightforward and simplified downstream development process that we have in place that involves clarification, coupling TFF with anion exchange chromatography. Obviously finalized with a sterile fill and an aseptic, sterile filtration, and then an aseptic filling. Just briefly, we see what a lot of folks in the field see when it comes to transition from an adherent culture to a suspension. Generally, we went through a very similar standard process of screening hundreds of clonal cell populations, doing dozens of rounds of transfection and looking at packaging across these various clones, identified a few key candidates, and then adapted those subsequently to suspension and serum-free culture. And it is now offered also to enable, similar to our plasmid offering, our 293 T cells are also offered to clients. And all of the components of our lentiviral platform do have drug master files with the FDA to help expedite your IND filing. We know this system well, and certainly what our lentiviral packaging cell line has shown is good growth behavior, able to achieve cell densities in the low 10 to the 7 cells per mil. And that our cell bank maintains viability above 90% through at least 144 hours of culture. And what we saw is similar in terms of looking at titers versus our adherent compared to our suspension. Both qualified cell banks, either the adherent or the suspension in this case. And it really is project dependent, but you can see in general what we observe is a shift in production, achieving higher titers, roughly 5 to 10-fold across some of the projects that we've done this side-by-side comparison with. So we feel comfortable that we have identified a cell and qualified a cell bank that can support higher yields in terms of your project. Certainly, it tends to be GOI dependent, and so this is good to see that regardless of the project we looked at, we saw it trending in the right direction. Besides the cell line, obviously, we also have optimized to support our platform, the various cell culture conditions, as well as essentially the optimizing packaging conditions through the transfection evaluation. So we certainly, again, standard evaluated a number of culture medias. And that really was critical for us. And then look through, altering in a DOE setting, pH, RPMs, and dissolved oxygen. We evaluated multiple transfection reagents, the concentration of those reagents that are used, and similarly the plasmid and concentration and ratio of the various plasmids. What time we harvest, and as I indicated earlier, we're getting potential early interest in the addition of enhancers to, again, increase either total yields or transduction titers. And following at least, this is one recent example, after we looked at the post optimization, again, in this case, about a 6-fold increase. But this can be, essentially when we work with clients that offer access to our platform, the first step is going to be to plug, obviously, your gene of interest and transfer plasmid into our known optimized conditions. But we can certainly perform additional optimization around a specific transfer plasmid should that be required, and really given the same types of DOE that I described for this example. Not a lot of detail here, other than just to show you generally. I think it's relatively standard, but following depth filtration and benzenase, we go through a TFF to concentrate, and buffer exchange for an anion exchange chromatography. The final buffer exchange, again using a TFF with an ultimate. With a terminal sterile filtration through a 0.22 micron filter. So in addition to the manufacturing, we also offer a full suite of quality control release assays, standard fare, looking at all the in-house at all the various safety type assays are listed here. And I think also wanted to emphasize the what's not listed here and what ultimately is a need for clients, and that is potency assays. They're obviously custom, and we have an analytical team that can offer potency assays for lentiviral vectors, depending on your application, be it whatever the readouts may be. We have the ability to multiplex proteins in a cytokine release type assays, ELISA for specific activation markers, as well as mixed cell population assays for example, in the oncogenic space and applications where you're mixing your cell therapy ultimately with the target cell type carrying the antigen of your target. So long story short, we can do a lot of this in-house and certainly have the ability to help on the critical potency assay on a custom basis. Just as a high-level example, so you get a feel for our turnaround and general time estimates. In this case, what we laid out is we are producing your shuttle plasmid. Obviously, we have three-tiered plasmid banking construction listed here. We offer qualified strains in this case, but certainly we see examples where it's just a primary cell bank followed by a master. But really to lay out expectations in general, should you want the full suite of services, including any plasmid production and QC, any PD that may be required in your plasmid space, essentially making one batch of plasmid, purification of that, allows us to now be in a position to start any lenti PD because we'll have material to assess how your shuttle plasmid and gene of interest fits into our platform. And generally, there's not a lot of huge changes here, but really it's going to be, in our experience, but it's going to be gene of interest specific. And that would lead to the production of an engineering batch, the QC of that batch, and then a pretty standard production of one lenti, all the QC, and support on the stability study and IND filing. We have a team that can offer both CMC documentation and support for IND applications, and subsequently BLA should the program be successful. But in a nutshell, it's about a 5-month turnaround, which is certainly I think is a pretty important aspect of many of the small early stage companies that are looking to get to the clinic quickly. I feel this kind of turnaround is impressive, and it's something that we take pride in and aggressively pursue with all of our opportunities in the lentiviral platform. I just wanted to put, since we're introducing our Center of Excellence in cell therapy also in Germantown, just to show our general workflows that we'll have there. This is really it's really focused on really an autologous cell therapy flow here. Obviously, this is also kind of standard fare for a CAR T, but you're in a situation where much of the cell therapies out there that involve gene modification are going to source somewhere, leukapheresis, or if it's healthy donor, et cetera. Enrichment of the target cell population, their activation and transduction with your lenti. Obviously, you're then removing your lenti or any gene modifier at that point, expanding the cells to a specific dose, harvesting and formulating as well. And cryopreservation if needed. I should also add here, it reminds me that we have spent a lot of time in final formulation studies of our lenti, and we have identified a formulation that tends to extend stability as well. It's something we can discuss with our clients should that be something that they are also of interest in pursuing. And then certainly just at a high level, when we interact with early stage clients, academic research grade processes, that general manufacturing considerations we have, and this is listed out on this slide, but it could be for really any platform offering or the Lenti I just described, is that if you're transferring a process, obviously it's an academic research grade process, and we have a lot of experience optimizing protocols to incorporate better process closure, for one example, and then obviously keeping in mind the end goal, which is to ultimately make this process GMP ready in an efficient fashion. And then the key data points that we also do in process evaluation is, again, at a high level, the target cell population in your starting material. If this is obviously with autologous, there's varying disease burden between sources. Wouldn't be a situation if you're sourcing from a healthy donor, say for an NK CAR, but obviously how many cells are there to start with, how well do we enrich at the time of gene modification, how well does that transduction take place, this transduction efficiency, how many total cells do we determine have been transduced, and then ultimately a determination of how long it takes to expand after transduction to meet the minimum requirement to meet all the QC needs, as well as to support the required doses for the clinical trial. So I'll summarize briefly. We bring a lot to the table when it comes to lentiviral vector production from, I think, highly efficient turnaround times in general in all of our platforms. But in the case of a lentiviral vector, we're in a position to offer access, as I said, to bacterial strains, to 293 T cells that are involved in. We have both constructed and fully qualified these bacterial and mammalian cell banks. We can support process and analytical method development in this space, whether it's your plasmid or your lenti, and we can do that through the listed approaches there, either focusing on upstream or downstream. In general, our PD tends to be on the upstream, since our downstream has shown to be pretty reproducible across various client vectors. It's the upstream and meeting the yields that they're looking for. Downstream, but we can offer that. And then certainly if there's anything custom with your program. And importantly, analytical development and qualification. And then, of course, once we're now into GMP production, we can make plasmid and lenti. We have a full suite of quality control tests that we offer and can validate methods as well, and support both release and stability studies. A lot of experience in designing stability plans for lentiviral and plasmids as well. We have two options depending on where we fill, but in general, we have the option of both fully automatic aseptic filling and a semi-automatic aseptic filling involving a pump, essentially, with an operator. And importantly, we can support all of your regulatory submissions, starting at pre-IND communications and the various agencies internationally, including the FDA's format submission documents, and then regulatory support for products that are also under research. So thinking ahead and not plowing ahead and doing all your development with unqualified materials. Obviously, we want to begin to add qualified assays and materials as early as we can in any kind of process development. Excited about Germantown, and certainly our highly trained and highly experienced teams that are supporting our cell therapy Center of Excellence through manufacturing of plasmids as well as lenti. Obviously, we have a lot of capacity when it comes to both plasmid and lenti. And the brand new shop in Germantown is eager to bring on clients, and we would certainly love to and anticipate presenting to potential clients a full description of the new facility in the near term. But I think in general, we have the ability through our various suites to really streamline your gene modifiers to get you into the clinic on the cell therapy side. And given our redundancy and flexibility in various sites, I think it also helps to set us apart in terms of the ability to have things running in parallel to early tech transfer on the cell therapy side while we generate qualified materials to support early phase clinical material. So I'll stop there. I had to show the slide with our new marketing bridge, since we take a lot of pride in being able to a bridge. It's a little play on uBriGene, and it's well thought out marketing campaign that I wanted to include to show that we really have the capability to help you from early stage research, preclinical activities to GMP readiness and clinical manufacturing. We do have commercial capabilities in a number of our sites. We also can bridge international markets. I think importantly, and something we also will probably provide a webinar in the future on, is how we can accelerate your clinical development programs through investigator-initiated trials in China, as well as the manufacturing turnaround times and cost can really expedite your full clinical development. And we bring to the table both manufacturing and testing. Obviously, it's important to offer a lot of this in-house, particularly when it comes to cell therapy, given the requirements of rapid methods. And we have those in place, as well as a talented team that can help with any analytical development that is more of a custom basis. So with that, I'll thank you all for your attention. Appreciate you finding time on your lunch hour to join, and we'll look forward to taking any questions you may have. Thank you.
Thanks so much, Ken. You nailed that half-hour slot. So let's go ahead and jump into some of the Q&A. I just want to invite everybody to submit their questions, their comments into the Q&A box over there to the right. But we have had a few that have come in. One, Ken, was, obviously this webinar is focused on lentiviral vector technology and our capabilities. What are kind of when you think downstream about the ultimate goal of an engineered CAR T therapy, what's the advantage of using lentivirus in cell therapy engineering with CAR T and CAR NK and various other genetically engineered cell therapies?
Yeah, good question. I think lenti will hold its place as a lead genetic modifying moiety for CAR Ts and cell therapies for a number of reasons. We're getting better and better as multiple generations come out in terms of the safety profile. And there's already a pretty extensive clinical track record. So there's the approach when you think about it is why reinvent the wheel? But then again, your lenti may not be the best at transducing your cell type. Certainly, gamma retros have a place there in certain cell types that aren't transduced as readily with lenti. But how we genetically engineer these cells for cell therapy applications is certainly an area of active investigation. And from gene editing approaches to RNA approaches, I think ultimately there'll be a place for those as well as new therapies that come online. There's also, I think, a drive to move, this type of, at least for cell therapy applications, to try to drive. I think developers are looking to drive this towards point of care. So that certainly is try to deliver basically the gene modifier to the patient's population at the bedside and reinfuse right into the patient in order to allow cell expansion to take place there. I think that's the power. It certainly would address a lot of the logistics in terms of I got a lenti coming from over here. I got the patient's cell sample coming from here. I'm manufacturing the cell therapy here, so it becomes a logistical nightmare. But in this case, I think that lenti can still hold its own there. but there may be a move towards some of the more, easily, more readily manufactured type gene modifications in that setting. But I do know of groups that are developing the point of care, still using Lenti as their delivery vector of choice. So, but I think, again, what's driving it will be addressing insertional immunogenesis of the Lenti, the cost of making the Lenti versus some of the more straightforward, say, RNA approaches, or something that would not have as significant a cost and wait time. I think ultimately there's going to be room for a lot of these gene modifiers, but Lenti is really, I think, leading the field.
Outstanding. Thanks for that thorough response, Ken. Another question, from someone regarding, the ultimate downstream application of these lentiviral vectors for CAR T. Question: for an autologous CAR T program, what's the typical scale of lentivirus production that will produce enough vectors to get your CAR Ts engineered for an IND enabling study, for example?
Yeah, again, I think I drove the message home quite a bit through the presentation that it really depends on your gene of interest. If we're achieving titers that are really high or in our standard kind of expectations, that would change. But our, standard, it's going to depend on how many patients you're going to want to treat, et cetera. But we've had a lot of interest in essentially the 20 to 50 liter batch size. Ultimately, that's what we've been quoting on for specific clients. But there's a lot of moving parts there in determining what it needs to be. But what we see, a majority are in the 50 liter range.
Outstanding. And we have the scale and the agility to be able to adapt depending on the titer and the gene of interest and whatever wrinkles.
Yeah, I mean, we have capability to do 200 liter runs, and we do have a 2,000 liter reactor. I think it would be unlikely that you would need to make 2,000 liters of Lenti, but certainly 2,000 liters of AAV is not out of the question. We have that capability, but yeah, that's
outstanding.
Kind of our standard package is 50 liters.
Thanks, Ken. Another question about the challenge of, lentivirus infectivity and losing infectivity during manufacturing. What is the uBriGene team? What sort of steps, without sharing any secret sauce information, but what do we do to be careful about maintaining infectivity throughout the manufacturing process?
It's all secret sauce. I can't answer that.
All right.
No, I would just say one thing. There's no secret sauce. I mean, we move quickly. We're working under conditions we've identified that don't have huge impact on titer, but ultimately Lenti is a fragile virus. So that's why we spent a lot of time looking at formulation for cryopreservation, because that's where you're going to take your biggest hit in our experience in terms of a freeze thaw. But certainly you lose a portion along the way. I don't. Certainly that'll evolve as additional technologies come on. But what we've identified for our downstream purification is what we've seen as the less destructive on the envelope vector and gives us the titers you need for a full CAR T program at a 50 liters.
Terrific. And another question that just came in a couple minutes ago: how big of a transgene can Lenti deliver, and is it possible to deliver polycistronic transcripts?
Well, it's got about a 9 kb packaging capacity. I'm sure you can find other potential, those that are focused more on developing the Lenti technology. And yeah, I don't see why you can't have multiple genes if they all fit, with the polycistronic approach, if they all fit within that packaging limitation.
Terrific. Anybody else want to submit a question? We still have a few minutes left, but I think I have covered all of them at this point. All right. Well, want to thank everybody for taking time out of the middle of their Thursday to join us. At least on the East Coast, it's the middle of the day. But from wherever you are, thanks for giving us your time and being generous with your time. We will be following up, asking if you'd like to have a more in-depth discussion about any of our capabilities, including the lentivirus platform that Ken reviewed today. Also in that email, we'll be letting you know that this is going to be posted, I think, on YouTube. So if you want to view it again or share it with a colleague or a friend to view, we encourage you to do that. And we look forward to having you join us for a subsequent educational uBriGene webinar. I think from talking to our marketing team, we're going to take a break during the summer since so many people are on holidays and enjoying the warm weather with their kids. So we'll probably pick this up again in September, is my understanding, and then have that monthly cadence again starting in September. So anybody has any questions, feel free to reach out to Ken or I or the generic inquiry email that was there at the on Ken's closing slide. And we look forward to learning more about your programs and how uBriGene can help you execute on those programs. All right. Thanks, everybody. Have a wonderful rest of your day.
Thanks, everyone.